Hello and welcome to another episode of the Science on the Edge podcast. I'm Dr. Mike Darcy, and in today's episode, I'm going to be performing a deep dive into the subject of cell death, specifically a form of cell death known as apoptosis. Now, this is something I've performed quite a bit of research into. Apoptosis was quite a core component of my PhD, and I've also published quite a successful paper in Cell Biology International on cell death. Specifically, apoptosis, necrosis, autophagy, pyroptosis, and a couple of other forms of cell death. Now, as I say, I'm going to be focusing today on apoptosis, which is the best understood form of cell death. So why is this such an important topic? Well, first of all, our cells are constantly being bombarded by damaging compounds in the environment. They are also being damaged by radiation, metabolic byproducts within our cells damage our DNA. So we accumulate DNA damage throughout our lives. Now, our cells try to repair this damage, but that's not always possible. And when the damage cannot be repaired, quite often a process known as apoptosis is initiated to cause a cell to go into a kind of programmed cell death to remove itself from a population. Failure to do so can result in cells that misbehave, that do not perform the normal functions, or that even become cancerous. So it's a very important process. Research into aging and longevity focuses quite strongly on trying to regulate how a cell repairs itself and when and if it goes into apoptosis. Okay, so if you are interested, by the way, in my podcasts, you're getting something from them, then it would be great if you donated a little to help me keep going with them. To do that, you simply go to scienceontheedge.com and click on the donate button. You can donate as little as you like. Okay, with that said, let's now dive deep into this topic. So here we go. Okay, so let's get started. So I'm going to be talking about a couple of different forms of cell death, apoptosis, autophagy, and a little bit of necrosis, but I'll mainly be focused on apoptosis. So what does it do? Well, in metazoans, such as us, apoptosis is vital for normal development. So as we go from being a single cell, a zygote, through being an embryo to a fetus to a fully grown adult, it's not just about making new cells, it's about getting rid of old cells. A hand starts as a stub, a big lump of cells. We don't grow fingers. What happens is we remove tissue to chisel out the fingers. And this is the case throughout our body. Our body is sort of like a lump of clay when we start. And the process of apoptosis is almost like a sculptor removing clay to shape the body. So we need this process of cell death known as apoptosis to allow our bodies to form during development. If apoptosis doesn't perform properly, our bodies fail to develop. When we are developed, we also need apoptosis to maintain homeostasis, to maintain the right number of cells in our body, to get rid of old and damaged cells. We also need apoptosis for our immune system to function properly. Its disturbance is implicated in numerous pathological conditions, ranging from degenerative disorders to autoimmunity to cancer. So a very important process. In animals such as the frog, you see apoptosis in a kind of macroscopic level. When a frog is a tadpole, it has a tail. The tail goes away, doesn't just fall off. What actually happens is it's absorbed. The tail, the cells of the tail enter apoptosis, the cells shrink, the components of the cells are broken down and they are recycled by neighboring cells. That's how you go from a tadpole to a frog. Okay, so apoptosis, necrosis, and autophagy. I'm going to be focusing on apoptosis in this talk, but I will touch on a couple of other forms of cell death too. So first, I'll give you a sort of general overview, then I'll dive deep and go into the nitty-gritty. There will be quite a bit of technical stuff in this talk. So first of all, how do we recognize apoptotic cells as opposed to cells that have died or are dying by some other mechanism. Well, there are certain characteristic biochemical changes in cells that undergo apoptosis. First of all, during apoptosis, you get chromosomal DNA cleaved into fragments, which basically means certain enzymes chop the DNA in the cells up into even pieces. You don't get that with a form of cell death such as necrosis, for example. So it's something that is very specific to apoptosis. You get a change in the plasma membrane, the membrane of the cell. A molecule known as phosphatidylserine moves from the inner membrane of the cell to the outer membrane. And this can be detected with certain chemicals and certain processes such as flow cytometry. There's a loss of electrical potential across the inner membrane of the mitochondria. Normally in a mitochondria, you have a positive charge inside compared with outside. That charge is lost during the process of apoptosis because the mitochondria becomes leaky. There's a relocation of a particular enzyme. Well, enzyme is quite the right word in this process, but let's just say molecule, known as cytochrome C, from the intermembrane space of the mitochondria to the cytosol. That's the cytoplasm of the cell. So cytochrome C is usually inside mitochondria, which are basically the organelle in the cell that generates all the energy. But during apoptosis, this particular molecule, cytochrome C, leaves the mitochondria and it can be detected in the cytoplasm, the liquid of the cell. So this is how we detect it. Now, before I go into apoptosis, I'll just briefly mention necrosis. So apoptosis is controlled, programmed cell death. This means that a series of processes are switched on, allowing for the cell to shrink, to break its components down into small membrane-bound vesicles that can then be safely absorbed by neighboring cells without collateral damage to surrounding cells. Now, sometimes it's not possible for a cell to go into apoptosis. The shock to the cell, the damage is just too severe. In such cases, a cell may undergo necrosis. So in necrosis, a cell gets damaged by some big insult such as heat or lack of oxygen or some chemical, and the cell simply swells up, becomes leaky, and bursts. So it swells up, known as oncosis, and pops, necrosis, spilling its contents onto surrounding cells. Now, this is not a good way for a cell to die because when it spills its contents onto surrounding cells, some of those contents are various digestive enzymes that are contained in the cell, and they act on neighboring cells, damaging them too. So in necrosis, it's not just the cell itself that's damaged, neighboring cells are damaged too. In apoptosis, however, which by the way is divided into two pathways, we have a controlled form of cell death. Now, first of all, I'm going to delve deeply into this, but before I do, I will just mention that apoptosis, broadly speaking, is divided into two pathways. There's more than two, but broadly speaking, two pathways. These are the intrinsic pathway of apoptosis and the extrinsic pathway. Now, to summarize what that means, the intrinsic pathway is where a cell realizes it's damaged. There are various sensors inside cells, such as the p53 protein, which detect damage. And when that damage is detected, the cell can be triggered to, first of all, try and repair itself, but failing that, if the damage is too extensive, to initiate apoptosis. That is known as the intrinsic pathway. And it involves basically various enzymes known as caspases, which I will talk about later. Then there is the extrinsic pathway. Sometimes the cell does not realize it's damaged, but luckily we have a second detection system. We have a type of immune cell known as a natural killer cell, and these cells roam around the body and they sort of sniff individual cells to see if they are healthy. And if these cells look damaged or infected in some way and need to be removed, then the immune cells will trigger the cells to go into apoptosis. This is known as the extrinsic pathway. It still involves caspases, the enzymes that trigger apoptosis, but a different group of caspases. So basically, intrinsic pathway is when a cell realizes it's damaged and triggers apoptosis, cellular suicide, all by itself. The extrinsic pathway is when an immune cell detects that the cell is damaged and triggers it to go into apoptosis. Then we have autophagy. Now, autophagy is a different process. Autophagy is a process that can be initiated by a number of different stimuli. For example, maybe there are not many nutrients in the environment, so the cells are at risk of starving, at which point the cells enter a process known as autophagy, where a number of enzymes are sort of activated that trigger the cell to sort of digest itself. No, I don't mean it kills itself, at least not always. I mean it tries to digest and recycle any damaged proteins in the cell, any damaged organelles. So anything that's in the cell that can be reused gets digested and recycled. Now, this can be a very good thing because as we get older, our cells accumulate lots of damaged proteins, damaged molecules that can cause the cells to misbehave and even become cancerous. And autophagy helps us to remove all of these damaging components. But if autophagy goes to its limit, if there is too much starvation, too much stimuli, autophagy can trigger apoptosis. So autophagy is sort of one of these mechanisms that can be very good for the cell, allow it to survive harsh conditions and even to become healthier. But if taken to its limits, the result can be apoptosis. The way autophagy works, by the way, in a very simplistic way, is there are organelles in the cells known as lysosomes. These are the stomachs of the cells. They contain lots of digestive enzymes. Their function is to help, recycle damaged components of the cells. In autophagy, these lysosomes fuse with various damaged organelles and structures in the cell, digest them, recycle them. That's basically how autophagy works. By the way, going back to necrosis, when a cell swells and pops, the damage to neighboring cells is caused to a large extent by the digestive enzymes from the lysosomes spilling out. Another reason why necrosis can be a bad thing. Okay, so that's an overview. Now I'm going to go into a deep dive. This is going to be quite technical. There's a lot of biochemistry in here. So you may want to pause every now and then just to get some of these concepts in your head, because I won't be going back, at least not much. Okay, but I'll try to take it carefully. Okay, so first of all, apoptosis depends on an intracellular proteolytic cascade that is mediated by caspases. What does that mean? Proteolytic means digesting proteins. Caspases are an enzyme, a group of enzymes in the cell that basically, when activated, can digest various proteins. They can break them apart and initiate apoptosis, so they can digest the cell. Now, we have these caspases in our cells all the time, but they're in an inactive form known as procaspases. So we have these procaspases. Think about a pen with a lid on it. If the lid's on, you can't write with a pen. Caspases have got the lids on in a default state. When there's a stimuli, either the intrinsic pathway or the extrinsic pathway, the lid is taken off the pen. The caspases become active. Now, there are two groups of caspases. There are the initiator caspases and the executioner caspases. When apoptosis is triggered by some sort of stimuli, the first group of caspases that are activated from the procaspases are the initiator caspases. So the initiator caspases become active. The lid is taken off the pen. These initiator caspases then take the lids off the pens of the executioner caspases and therefore activate them. And it is the executioner caspases that break the cell down and cause apoptosis. So these caspase enzymes are very important. They exist in the cell as inactive procaspases. When they are activated, they become caspases. First, the initiator caspases are activated, and then the initiator caspases activate the executioner caspases, which cause cell death. Okay, so we're going to go through the two main pathways now. First, the extrinsic pathway. As I mentioned, the extrinsic pathway of apoptosis is when a cell is told to go into apoptosis, is triggered to go into apoptosis by a natural killer cell, a killer lymphocyte. So a circulating immune cell that notices the cell is damaged and triggers it to go into apoptosis. So for those of you watching this on YouTube rather than listening to it through a podcast app, I've got some diagrams here that I'm going to explain. I'll try to explain them in a way that you can get even if you're just listening to this podcast. Okay, so first of all, I have a diagram present here where I have a simplified cell with just the molecules listed inside it that are involved in the extrinsic pathway of apoptosis. And I also have a diagram of a killer lymphocyte. And I'm going to talk about the process now whereby the killer lymphocyte stimulates the extrinsic pathway of apoptosis. So first of all, the killer lymphocyte, when it detects that a cell is damaged, and I'm not going to go into how it does that right now, but it involves something called MHC, major histocompatibility complex. Basically, a cell displays bits of itself on its surface, and if those look damaged in some way, then the killer lymphocyte realizes the cell is damaged and should go into apoptosis. So first of all, the killer lymphocyte has on its surface a ligand, which is kind of like a key. Specific kind of ligand known as a Fas ligand. Now, when the killer lymphocyte decides it's going to initiate apoptosis in a target cell, it lines up its Fas ligand with a Fas death receptor that's on the surface of the target cell. And that fits like a lock and a key. The Fas ligand fits with the Fas death receptor. Now, the Fas death receptor has what we call an extracellular domain, meaning part of it is on the surface of the cell, and an intracellular domain, meaning part of the protein is inside the cell, poking into the cytoplasm. Now, when the Fas ligand binds with a Fas death receptor, this activates that receptor. And the inner domain of the Fas death receptor is known as a death domain. So that bit of protein that sticks in the cell is known as a death domain. When the Fas ligand plugs into the Fas death receptor, the death domain is basically activated. Now, floating inside the cell, there are lots of proteins known as Fad adapter proteins. These Fad adapter proteins also have a death domain. So their death domain can stick to the death domain of the Fas death receptor when it's activated. So the end result here is all these Fad adapter proteins sort of line up to the activated Fas death receptor and stick their death domains against the death domains of the Fas death receptor. Now, the Fad adapter protein doesn't just have a death domain, it also has at the other side a death effector domain. So these death effector domains stick to other death effector domains when activated. Another thing that has a death effector domain are the procaspases. These procaspases that are involved in the extrinsic pathway, so procaspase 8 or 10. They again are just floating around in the cell. But when the Fas ligand binds to the Fas death receptor, which activates the death domain of the Fas death receptor, sticks to the death domain of the Fad adapter protein, lining everything together, the procaspases stick their death effector domain against the death effector domain of the Fad adapter protein. Long story short, everything sticks together. It's basically what we've got. We've got a big complex stuck together, Fas ligand to Fas death receptor, death domain of the Fas death receptor to death domain of the Fad adapter protein, death effector domain of the Fad adapter protein to death effector domain of the procaspase 8 or 10. The whole thing comes together into a complex known as a disc or a death-inducing complex. So what we've done now is we've brought lots of procaspases together in this disc. They're all stuck together. Now, when you bring procaspases close together, they react with each other, and the pro part is chopped off, meaning you go from a procaspase to an active caspase. So now we have activated initiator caspases, in this case, caspase 8 and 10. They will then activate the executioner caspases, which will then cause the cell to go into apoptosis, to shrink and to break apart. So this is the extrinsic pathway. If you didn't get that, maybe just wind back and go through it one more time. I'm now going to talk about the intrinsic pathway of apoptosis. Now, the intrinsic pathway, as you recall, is when a cell realizes it's damaged and initiates apoptosis itself. So the intrinsic pathway of apoptosis, it basically is dependent on the mitochondria. These little machines, these little organelles in our cells that are responsible for making energy in the form of something called ATP. So the mitochondria help trigger this intrinsic pathway. Okay, so for those watching this on YouTube, what we're looking at is a bunch of molecules that are floating around inside the cell. So this is all going on inside the cell. So the intrinsic pathway works. So we have the mitochondria. Within the mitochondria, we have a particular molecule known as cytochrome C, which lives inside the mitochondria. It's involved in respiration. It normally only exists inside the mitochondria. But if the mitochondria gets damaged, gets holes in it, the cytochrome C leaks out into the cytoplasm of the cell. But normally it would not be present in the cytoplasm of the cell. Okay, so involved in the intrinsic pathway are the mitochondria with cytochrome C. We have another molecule that's floating around in the cell, many copies of it, known as APAF1. The way to think about APAF1 is it's kind of like a closed hand. In the middle of the hand, not exposed to the rest of the cell, is something known as a card domain, which actually stands for Caspase recruitment domain, but it's not exposed in a healthy cell. Now, also floating around the cell, we have another procaspase, the procaspase involved in the intrinsic pathway. This is known as procaspase 9, which also has a card domain. Card domains bind to card domains. The procaspase 9 can't bind to the APAF1 card domain at the moment, though not in a healthy cell, because the card domain is not exposed, it's closed. APAF1 is folded in on itself. Then we get an apoptotic stimuli. The cell is damaged. It's detected by various molecules such as P53, known as the guardian of the genome. I'm not going to go into the process of that right now, but let's just say there are lots of sensors in the cell. When these sensors detect the cell is damaged, they try to repair it. They activate various genes involved in cellular repair. When that fails, various genes involved in apoptosis are activated and various proteins are produced that trigger apoptosis. I'm going to get into that shortly. So there is some damage to the cell. The intrinsic pathway is activated. What happens? Well, the proteins involved in the intrinsic pathway cause holes to form, channels to form in the membranes of the mitochondria. This allows cytochrome C to leak out of the mitochondria. The cytochrome C then binds to the APAF1. And when it binds to APAF1, remember the closed hand, the hand opens, exposing the card domain. A bunch of APAF1s then stick together. All the card domains stick together and they form a kind of windmill structure. So a windmill of APAF1s with all of their card domains stuck together in the middle. Okay, now as I mentioned, card domains attract card domains. So this windmill attracts procaspase 9. It attracts procaspase 9. The procaspase 9 card domains bind to the card domains in the center of the APAF1 windmill. By the way, this windmill is known as an apoptosome. Now, what we've done now is we've brought lots of procaspase 9s together into close proximity. Remember what happens when we bring procaspases together? They react with each other and the pro part is chopped away. So now we have active caspase 9. Caspase 9 is an initiator caspase, so it now activates the executioner caspases and apoptosis occurs. Okay, if that wasn't clear, please rewind, go through it again. I'm now going to move on to a little bit more detail. It's even more complicated than this, unfortunately. But it happens all the time in our bodies. Okay, so which proteins are actually triggering the apoptosis in the intrinsic pathway? Well, a class of proteins known as the BCL2 proteins. The BCL2 family of proteins are involved. The first one that was discovered was a protein that was named BCL2, but then many other proteins were discovered that were very similar to it. So a family was named. So BCL2 is the family of proteins, and it's also one of the proteins, the first one that was discovered. Now, the BCL2 proteins, they come in sort of three categories, but two broad categories. First, there are the anti-apoptotic BCL2 proteins. They include BCL2 itself and BCL-XL. So these are proteins which try to stop apoptosis. You don't want apoptosis to just happen all the time. You only want it to happen when the cell is damaged. So we have these anti-apoptotic proteins such as BCL2 and BCL-XL that prevent apoptosis. They keep the cell alive. Then we have the pro-apoptotic proteins. Now, these are known as the BH123 proteins. These include proteins such as BAX and BAC, and the BH3 only proteins, which include many proteins such as BAD, BIM, PID, PUMA, and NOXA. Now, I'm throwing a lot of information at you now, so I'll just break it down. Basically, the BCL2 proteins all contain 4 domains in the protein. A domain is just a region of the protein. They contain the BH1, BH2, BH3, and BH4 domains, and they are anti-apoptotic. They basically stop a cell from dying. Now, the other two groups of BCL2 proteins, the ones that are pro-apoptotic, they come in two categories. BH123, which is a protein such as BAX and BAC. They are called BH123 because they have the BH1, the BH2, and the BH3 domains, but no BH4. And these are pro-apoptotic. Then we have the BH3 only proteins, BAD, BIM, BID, PUMA, NOXA, etc. They're called the BH3 only proteins because the only domain they have in common with the rest of the family is the BH3 domain. So we have pro-apoptotic BCL2 proteins, and we have anti-apoptotic BCL2 proteins, pro and anti. So how does this work? Well, in a normal cell, a cell is in a kind of homeostatic balance. There are proteins saying grow, divide, and there are other proteins saying stop growing, repair, maybe even go into apoptosis and die. And they're in a sort of balance in a typical cell. Now, first of all, we have those BH3 only proteins. The BH3 only proteins. So these are proteins, just zoom back again so you can sort of see. These are the BH3 only proteins. These are BAD, BIM, BID, PUMA, and NOXA. So they are the BH3 only proteins. Then we have the BH123 proteins, which are BAX, BAC, BOC, those sorts of proteins. Okay, so the BH3 only proteins, PUMA, NOX, all of these, they stimulate the BH123 proteins, such as BAX. I'll focus on BAX in this case. And then BAX initiates apoptosis. How does that work? Well, I'll talk about that later in a moment. But the end result of its pathway is initiator caspases get activated. At the same time, the BH3 only proteins, such as NOXA and PUMA, etc., are inhibiting BCL2, the anti-apoptotic protein. BCL2, if there's enough of it, will inhibit BAX and will also inhibit the initiator caspases. So we've got BCL2 trying to stop apoptosis, stop the BH3 only and the BH123 proteins. And we've got the BH3 only trying to inhibit BCL2 and stimulate BAX. And if there's whether a cell goes into apoptosis or stays alive depends on the balance. If there's more BH3 than BCL2, cell will go into apoptosis. If there's more BCL2 than BH3, the cell won't go into apoptosis. So it's a balance between the two. It's a homeostatic balance, which is broken by damage to the cell. So what happens during apoptosis then? The intrinsic pathway, I should say. So here we have the mitochondria. And now the mitochondria has lots of inactive BH123 proteins in its membrane. These are proteins such as BAX. So we'll just talk about BAX. So there's lots of BAX, which is a BH123 protein in the membrane, but inactive. When there is an apoptotic stimuli, the BH123 proteins come together and form channels through the membrane of the mitochondria, which lets the cytochrome C out and causes the intrinsic pathway to be activated as discussed previously. Now, if apoptosis is inactive because there's too much BCL2, this is how it works. If you've got lots of BCL2, the anti-apoptotic protein in the cell, lots of it, it basically holds on to the BH123 protein, so the BAX. It holds on to it and it stops it from coming together to form those holes, those channels in the mitochondrial membrane. So if you have lots of BCL2, apoptosis can't happen because those channels cannot be formed by the BH123 proteins, and the cytochrome C cannot leak out, where it would bind to the APAF1 and initiate apoptosis. Now, if, however, there's lots of the BH3 proteins present, such as purine oxide, etc., then what will happen is it will grab hold of the BCL2. It will actually grab hold of it and stick to it. And if it's holding on to the BCL2 protein, the anti-apoptotic protein, then the BCL2 protein cannot then grab hold of the BH123 proteins. So the BH123, such as BAX, is now free to come together to form those channels in the mitochondrial membrane, leak the cytochrome C out, and initiate apoptosis. Okay, now we have some other molecules involved in this process, which I'm not going to talk about much, but we do have them. We have a molecule which is normally present inside the mitochondria called an anti-IAP. An anti-IAP. I'm going to explain what that means shortly, but basically we have so normally this anti-IAP would be inside the cell, inside the mitochondria, and we have the IAPs sort of floating around in the cytoplasm. Now, the IAPs basically bind to caspase and sort of block it. So they inhibit apoptosis. They are inhibitors of apoptotic pathways, these IAPs. They bind to caspase and they sort of stop it. Now, when you've got the holes in the mitochondria, though, the cytochrome C leaks out, but also so do the anti-IAPs. And these anti-IAPs block the IAPs and make it more likely that apoptosis will occur. Okay, so that was a deep dive. Now let's sort of discuss in a sort of summary what's going on. So we have several different pathways of cell death. We have autophagy, which is really just about recycling components of the cell, but in times of need, when the cell is starving, for example. But autophagy, if it goes too far, can trigger apoptosis, which is programmed cell death. Broadly speaking, programmed cell death, apoptosis, is divided into the intrinsic pathway and the extrinsic. The intrinsic pathway, when the cell realizes it's damaged and therefore triggers apoptosis itself via the mitochondria. And we have the extrinsic pathway, and the cell doesn't necessarily realize it's damaged, but the immune cells do, and they tell the cell to go into apoptosis. That's the extrinsic pathway. We also have necrosis, which is uncontrolled cell death, which has some purposes, but generally is just causes damage. Too much apoptosis means cells will die too easily. And this has been linked with conditions such as Alzheimer's disease, Parkinson's disease, a number of other conditions where cells die a bit too easily, and generally reduced lifespan if you have too much apoptosis. However, too little apoptosis means you let cells get away and continue living, even though they're damaged. And this can result in cancer and various other conditions too. So nature sort of evolved a sort of balance between too much apoptosis, too little. For most of our lives, we keep that balance in check. The balance is held in check by those pro and anti-apoptotic enzymes, but genetically we're all a little bit different. Some of us have cells that go into apoptosis easier than others. Other people have cells that don't go into apoptosis so easy. There are benefits to either of those processes, really. But we do need apoptosis to remove damaged cells. So to conclude, apoptosis is extremely important. It's more complicated than I've just explained, by the way, but this is a good overview. I may revisit the subject at a later point. For any scientists out there or anyone that wants to delve deeper, I do have a paper on cell death, a review of the major forms of cell death published in Cell Biology International. You can look that up online. And in that paper, I go into a lot of depth about the different forms of cell death. Okay, so thank you very much for watching or listening. If you want to know more about me, you can follow me on Twitter. My hashtag is @MarkOfTheD. That's MarkOfthe capital D. Or you can visit my website, which is scienceontheedge.com. And if you like, you can even make a little donation by clicking on the donate button. Okay, thank you very much for listening, and I look forward to speaking with you all again very soon. But goodbye for now. Goodbye.